Welcome, future colleagues, to this high-yield explainer. I'm Professor Roberts, joined by Professor Chen. Today, we're returning to the absolute fundamentals of microbiology, viral structure. A disproportionate number of questions across the NEET PG, INICET, and UPSC CMS examinations are built entirely on three structural pillars: envelope status, genome type, and virion-packaged survival enzymes. We're going to systematically break down this classification so you can predict pathogenesis and transmission routes purely from structure rather than relying on rote memorization. Here's our outline for today's drill-down approach. We'll start with core components and symmetry, then move to the crucial distinction of enveloped versus non-enveloped viruses. After that, we'll cover the Baltimore Genome Classification Scheme, virion-packaged survival enzymes, and finish up with a rapid-fire MCQ drill. Let's jump right into Section 1, Core Components and Symmetry. We'll analyze the fundamental architecture of a virus from the inside out. At the center, we have the viral genome surrounded by a protein shell called the capsid. Together, the genome and capsid form the nucleocapsid. The virion, by contrast, is the complete, infectious viral particle. Professor Roberts, we need to flag exam trap number 1 right here. Students frequently use the terms nucleocapsid and virion interchangeably. On the exam, that's a critical error. For a non-enveloped virus, the virion is structurally identical to its nucleocapsid. But for an enveloped virus, the virion includes the nucleocapsid plus the acquired lipid envelope. Precise terminology is absolutely paramount. Surrounding the genome is the capsid, which is composed of individual protein subunits called capsomeres. These follow specific architectural symmetries. We see icosahedral or cubic symmetry in adenoviruses and herpes viruses. We see helical symmetry in many RNA viruses, and complex symmetry, which lacks conventional geometry in pox viruses and bacteriophages. And this leads directly to exam trap number 2. Do not assume helical means non-enveloped. In medically important human pathogens like influenza and rabies, helical nucleocapsids are almost always enveloped. Never conflate capsid symmetry with envelope status. They are completely independent variables. Moving to Section 2, enveloped versus non-enveloped. This is the highest yield structural concept in this entire topic. Enveloped viruses possess an outer lipid bilayer acquired from the host cell membrane. This layer is studded with viral glycoprotein spikes and stabilized by matrix proteins. You might assume this extra layer provides extra protection, but the clinical reality is a paradox. Exactly. The virus requires this intact lipid bilayer for infectivity. Therefore, enveloped viruses are exceedingly fragile in the environment. They're susceptible to drying, heat, detergents, lipid solvents, and alcohol. Because of this, they typically require direct transmission routes that maintain moisture, like respiratory droplets, blood, sexual contact, or solid organ transplantation. Conversely, non-enveloped or naked viruses rely solely on their resilient protein capsid. They're highly resistant to drying, detergents, lipid solvents, and even the harsh acidic pH of the stomach. This immense structural resilience allows for successful transmission via the fecal-oral route and environmental fomites. This brings us to exam trap number 3, an extremely high-yield infection control point. Why do alcohol-based hand sanitizers routinely fail during norovirus and rotavirus outbreaks? The answer lies strictly in their structure. Alcohol functions as a disinfectant by disrupting lipid bilayers. Since norovirus and rotavirus are non-enveloped, they completely lack the lipid layer that alcohol targets. So in these specific outbreak settings, mechanical removal via soap and water hand washing is a strict clinical mandate. Let's move inward to Section 3, the Baltimore Genome Classification Scheme. Breaking down viral genomes by nucleic acid type, strandedness, and polarity reveals distinct categories. In the double-stranded DNA category, we have herpes virus, adenovirus, and pox virus. Crucially, in human pathology, the single-stranded DNA category is essentially represented by parvovirus. Looking at RNA genomes, the classic double-stranded RNA example is rotavirus. Single-stranded RNA is further divided by polarity. We have positive sense, such as picornaviruses and flaviviruses, and negative sense, which includes influenza and paramyxoviruses. Now, we must frame exam trap number 4, arguably the most vital genome rule for test day. Only the positive sense single-stranded RNA genome is directly infectious as isolated nucleic acid.
That's right. If isolated and stripped of its capsid and envelope, it's infectious on its own because it possesses the exact same polarity as host messenger RNA. The moment it enters the host cytoplasm, it can be translated directly by host cell ribosomes to synthesize functional viral proteins.
While rules are foundational, examiners prefer exceptions. Mark down exam trap number 5. Do not assume all RNA viruses are haploid. Retroviruses, such as HIV, unusually possess a diploid genome. They carry 2 identical copies of single-stranded RNA within the virion. And exam trap number 6. Do not misclassify hepatitis B virus. Students frequently identify HBV merely as a DNA virus and ignore its replicative quirk. HBV is indeed a double-stranded DNA virus, but it uniquely depends on the enzyme reverse transcriptase to replicate via an RNA intermediate. Now for section 4, virion-packaged survival enzymes. Certain viruses face a physiological barrier because the human host cell lacks the specific enzymes required for their replication. Therefore, these viruses must bring their own preformed catalytic machinery packaged inside the mature virion. Negative-sense single-stranded RNA viruses, as well as double-stranded RNA viruses, must package RNA-dependent RNA polymerase, or RDRP. Human cells simply cannot synthesize RNA from an RNA template. Similarly, retroviruses and hepatitis B virus must package reverse transcriptase to synthesize DNA from an RNA template. Finally, pox viruses must package DNA-dependent RNA polymerase. Because pox viruses uniquely replicate entirely within the host cell cytoplasm, they cannot access the host cell's nuclear RNA polymerase machinery and must supply their own. We conclude with Section 5, Rapid-Fire MCQ Drill. Professor Chen, I'll pose the clinical vignettes and you provide the single best answer rationale. Let's begin. Question 1. A patient contracts a viral gastroenteritis via the fecal-oral route. Which structural feature of non-enveloped viruses precisely explains their resistance to gastric acid and detergents? The correct mechanistic rationale is the absence of a lipid envelope. The protein capsid alone provides immense structural resilience. Without a fragile lipid bilayer to disrupt, lipid solvents, acid, and detergents are largely ineffective. This cements the core survival advantage of naked viruses in harsh environments. Question 2. If you artificially extract viral nucleic acid and inject it into a permissive host cell, which RNA genome polarity is directly infectious as isolated nucleic acid, and why? The answer is positive sense single-stranded RNA. The rationale is direct translation. It shares the exact polarity of human mRNA, which eliminates the need for prepackaged polymerases. The host cell liposomes immediately initiate protein synthesis upon entry. Question 3. This is a classic trap designed to catch students who categorize viruses too rigidly. Which DNA virus uniquely depends on reverse transcriptase during its replication cycle? Hepatitis B virus, a member of the Hepidavnovirus family. While retroviruses are RNA viruses utilizing reverse transcriptase, hepatitis B is the specific DNA exception to the rule. Never misclassify this replicative deviation. Final question. If a virus possesses a negative sense RNA genome, which specific enzyme must it package within the mature virion prior to infecting a new host cell? That enzyme is RNA-dependent RNA polymerase, or RDRP. The virus requires RDRP to generate a translatable positive sense intermediate from its negative sense template before any viral protein synthesis can commence.
Excellent. I'll leave you all with this concluding thought. When you analyze a virus in a clinical vignette, don't just see a name to memorize. Ask yourself, is it enveloped? What is its genome polarity? What enzymes must it carry? Mastering these structural pillars transforms viral pathogenesis into predictable logic for test day.
Thank you for joining this high-yield explainer. Keep reviewing systematically, stay focused, and best of luck in your medical examinations.